Well, as you know, our goal at this point is to begin to really peel back the layers on that onion, metaphorically speaking, of course, that we call hope. And we're going to take one more diversion en route to that goal. Namely, we're going to talk not about hope directly until the end of this presentation, but rather about this thing we call love. And we're going to explore a little bit about the biochemistry and neurochemistry of that emotion that we call love. And the reason for doing this is that humans have been fascinated by this concept for as long as we've had the capacity to record our thoughts and communicate those to future generations. We've agonized over it, we've celebrated it, we've pursued it. It's awash in our culture every moment of every day. You know, and if we look at the natural world, it's a little more sane in some respects. Because, you know, in nature, you got a mating system, you got a mating season, the males compete for the available females, everybody gets pregnant, and that's pretty much the end of the story. It's pretty perfunctory, it's need-based. You got to keep the species going, so you do what's necessary to make that happen, and all is well. But in the case of human beings, we've layered it with all sorts of complexities and other considerations, and innumerable subtleties that have preoccupied us when maybe we should be sleeping or working or doing something productive, but rather we're agonizing over this thing called love. And if you just think about its presentation among mankind, I think this becomes immediately obvious. I've selected a couple of films for us to discuss in just a moment. But before we get there, let me regale you with the reality that we're going to be looking at this system of behavior in 4 parts. We're going to talk, first of all, about the neurobiology of love as envisioned by Helen Fisher. No, she's not related to me. She's a fairly famous scientist. Secondly, we'll look at the 3 biochemical stages of love identified by Fisher. And specifically, we'll talk about the neurotransmitters involved in those 3 stages. Then we'll look at some really interesting experiments with a little animal called the vole and how it demonstrates the reality of what Fisher is saying about these neurochemicals. And lastly, we'll talk about the evolutionary significance of this thing called love and its relationship, finally, to hope. So, as I was saying moments ago, this thing called love is something that has been an intense focus of mankind forever. And because it is so well known, so well studied, so well romanticized, it is something that we grow up with. It's something that we are taught to seek in adulthood, and many of us will spend a lot of time in its pursuit. So it's not a trivial undertaking. It's not a trivial thing in our lives. In some cases, it's a profound and ongoing focus. And as I mentioned, it is the stuff of cultural icons, this thing called love. Our culture celebrates the craziness of the romantic part of love in every modality conceivable, but I've chosen here 3 examples drawn from the motion picture business, starting with the movie called Roxanne, in which Steve Martin plays this poor guy with this really big nose, sort of a Cyrano de Bergerac kind of guy, who is just odd looking and not the kind of guy you attach to romantic love. And in this movie, Steve Martin is asked by another guy to help him win the love of this woman named Roxanne. And in the process of trying to help this other guy secure the love of Roxanne, Roxanne falls in love with Steve Martin and his ugly nose, and of course this begets all sorts of chaos and feelings of betrayal by the friend and so forth. And it's just one step on the crazy, crazy, crazy ladder of love. But maybe it's the most understandable and the most realistic of the three movies I'm going to talk to you about. The second one is the updated version of the movie Titanic, starring Kate Winslet, who appears in this movie as a woman with a very secure future. She is brought up as an upstanding member of society. She is engaged to be married. She knows exactly what she's going to do with her life and how it's going to play out. And then, within minutes of getting on board the Titanic, she encounters, shall we call him, bad boy, Leonardo DiCaprio, who is very clearly from the wrong side of the track. You can just tell that from the way he dresses. It's just not upstanding like Kate is. But somehow, Leonardo manages to sweep Kate off her high-heeled feet. She falls for him in a big way, and before you know it, the two of them are, shall we say, in flagrante delicto in the backseat of a car in the cargo hold of the Titanic. That's how she lost her virginity. And you also know that this is not going to end well, and that by the end of the day, Leonardo's going to willingly give his life to save Kate, and that is how he goes down and how he demonstrates his love, which is all of 24 hours old at that point. So, how likely is that? 24 hours, he's already ready to die for her. And she is at that point ready to abandon her entire upbringing, all of the hopes and dreams that she had, as well as her parents. It's crazy. What can I say? And that craziness is certainly in evidence when we go on to our third film, the iconic Romeo and Juliet. There are many versions of this film. I like particularly the one with Len Whiting as Romeo and Olivia Hussey as Juliet. They are just beautiful, beautiful people, and they play the roles rather well. And you know how this is. They don't meet each other socially because they're, well, in the normal course of events, because their families hate each other. But they end up at the same dinner party together, and they see each other across the room, and bam, they're in love. Now, you got to wonder about that, but that's the way it's written. That's the way Shakespeare wants it to be. And so they immediately fall in love, and as you know, it does not end well for those folks, but they go to their graves willing to die for one another after having spent, at least in the movie version, one very special night together. So what is it that can impel us to do these crazy things that in the latter two cases involve mortality, and with very little preparation in terms of knowing the person for whom they're sacrificing? It's an amazing, amazing thing that we humans, who are otherwise rational, can be so irrational when it comes to love. And why is that? Well, here we need to get into the material with Helen Fisher, who is a legitimate scientist. She has published scientific papers in real scientific journals. I've given you one example in the slide. She published the paper mentioned here in the Journal of Neurophysiology, which is a pretty well-known journal in the sciences. And she was interested in defining the underpinning, the biochemical underpinning of the different stages of love. And while we could simply go to her paper and present some tables and graphs, I instead am reprising the material that she put into her popular book on the subject. By popular, I mean it's a book designed for non-scientists. And so she published this book called Why We Love, and that is the source of the material I'm going to be presenting to you in the next few slides. So what did she do exactly? Well, she recruited 40 people, young people, who were, by their own admission, madly in love with a particular individual. And she took these people and individually she put them in a magnetic resonance imager. It's a way to scan your brain. It's based on a huge magnet that has the ability, when you mess with it, to change the way atoms and molecules in your body send their signals, and this can result in an image which is then used to gauge the state of a brain in a particular moment under particular conditions. And the particular conditions in this set of experiments were that the 40 young people were put into the MRI machine, and they were asked to perform a series of tasks. The first was to look at a picture of the person whom they loved, and they loved with all their heart, mind, soul, etc. So they looked at their special person for 30 seconds, and all the while the MRI machine is snapping pictures of their brain in love. Then they were provided with a diversion task. That could be something like counting to 10 or asking what the weather was, something that was completely irrelevant to being in love, in other words. And then after that, a control was conducted in which each individual was asked to look at the photograph of an acquaintance, but not somebody with whom they were in love. And again, the MRI machine took pictures of their brain under those conditions. And the images mentioned were shown more than once. Every 30 seconds, a different image or task was performed, and this went on for a period of 12 minutes. So they had three basic things to do: look at the picture of the person they loved, do a diversion task, and finally looking at a picture of an acquaintance. And they cycled through these three things for 12 minutes. Okay, so when all was said and done, Dr. Fisher obtained what she called a photo album of love. And what she noted in particular was that when the subjects, the 40 recruits, looked at a picture of their loved one, what Fisher saw in the brain scans was massive amounts of the neurotransmitter / neurohormone dopamine. Dopamine was released in large amounts, and it appeared in the scan as an intense red color. So you see here a brain scan, and you see lots and lots and lots and lots of red. So this would be typical of what she found when the subjects in her experiment were looking at the picture of the person they loved. When they looked at the acquaintance, not nearly as much red. When they did the diversion task, no red. So we know that dopamine is probably important in that feeling of intense romantic love. We also know that it's dopamine has effects in various parts of the brain, but including the prefrontal cortex and lots of other places. It's made in lots of places. It has effects in lots of places. The midbrain is also loaded with dopamine receptors. So when dopamine is present in that amount, it's doing a lot of things. But in this case, there was a distinct uptick in the amount of dopamine when the people were shown the picture of the person whom they professed to love romantically. Now let's take a minute to talk a little bit more about this thing called dopamine. It's a small protein consisting of a couple of amino acids. It's an interesting molecule in the sense that it can act both as a neurohormone, which is what is supplying the romantic feelings that we've been talking about, but it can also act as a neurotransmitter. In other words, it can help neurons talk to one another. It can send the message and diffuse across the synaptic cleft and then start a new action potential in the next neuron. So this is the first such molecule that we're encountering. Dopamine is the first molecule that can play the role of both a neurohormone and a neurotransmitter. We distinguish between neurohormones and neurotransmitters in the following way. Hormones are materials that are produced and released by one part of the body, but then travel to some other part of the body to exert their effect. Neurotransmitters, on the other hand, are those materials that are released by the presynaptic neuron to send the message to the next neuron. Now, dopamine is one of these materials that can serve both as a neurotransmitter and a neurohormone, but others that are sort of related to dopamine chemically can also share this dual role, and they include the materials known as epinephrine and serotonin, both of which we will encounter in a couple of modules. But getting back to this thing called love, Helen Fisher says that love is not an emotion in the way we like to think about emotions. Rather, it is a motivational system or a drive that is part of a reward system. Her basic argument is that we need to keep the species going, and therefore we need to reproduce to help keep that species going. And she observes something that I've been talking about since the beginning, and that is that we are a slow-growing, long-lived species with a really big brain. And this means several things of importance, but one is that our babies are born in an immature state, something I will elaborate upon several times, and as a consequence of that dependence, we need to keep the parents together somehow long enough to help that child get through the dependent phase. And what Fisher says is that's why we have these chemicals that I've alluded to. In fact, she argues that we are virtual slaves to our neurotransmitters. We're basically unable to not do what they tell us to do. They, for instance, compel us to seek a particular lover, not just any random person, but a particular lover to become one's mate. The brain links this, that is to say, the specific person to specific emotions, and those emotions change over the course of a long relationship. We mentioned the prefrontal cortex earlier. It is activated by dopamine, and it has a lot going on in the case of romantic love. It's busy assembling data. It's putting information into patterns. This is particularly behavioral information as you interact with the potential beloved one. Also plotting strategies. If you've decided on a particular person, there's a fair amount of strategizing. What if there's a boyfriend or a girlfriend already? What are you going to do? All of these things get monitored as progress towards the goal, namely securing true love. And Fisher identifies 3 specific stages of love, each of which has a very different neurochemistry associated with it. In the first stage, she calls it the lust phase, the person undergoing lust craves sexual gratification. And this is driven by the sex hormones, primarily testosterone if you're a guy, and the estrogens if you're a woman. Next, assuming you've found a person, the next phase is the attraction phase, which we also know as romantic or passionate love. And this is characterized when you're in it by a feeling of euphoria when you're in the presence of the one that you love. We know that this is mediated by the amygdala, which has responsibility for sensations that involve pleasure. So chocolate has its effect here, cocaine has its effect here, and romantic love has its effect on the amygdala. One also finds some traits arising during this period that are not necessarily good for you, but but nonetheless occur as part of romantic love, including obsessive thinking, pretty much like obsessive compulsive behavior. And again, it's driven by high dopamine levels and, as it turns out, low serotonin levels. Lastly, we get to the attachment phase of love, in which the crazy, passionate, romantic love is replaced by a sense of calm and peace and stability. You've reconciled yourself to your lifetime partner. You've learned to put up with the fact that he leaves his socks in the floor or that she squeezes the toothpaste from the middle of a tube. You don't really care anymore. It's all good, and life goes on. And this, of course, also is driven by particular neurohormones. In this case, oxytocin has a major role, and to a certain extent, vasopressin. And look at this happy couple. They look like they're in the calm, peaceful, stability stage of a relationship. So why is all this necessary? Why do we need all these very powerful hormones to impel us to show certain behaviors? Well, it makes sense when you think about how uncomfortable going out to find a mate is. When you're alone, when you're looking for a partner, you need really powerful hormones like testosterone, like estrogen, to get you out there on the prowl. There's a certain amount of anxiety that goes with seeking a relationship, and in order to get beyond that anxiety and get out there actively looking for somebody, it helps to have these powerful hormones behind it. When you get to the romantic attachment phase, the goal from a biological standpoint is to focus on a single individual and have a pair bond form between those two individuals, so that when we get to the child rearing part, which takes a number of years, the feeling of romance keeps the two bound together, and they stay together long enough to raise that child to a state of independence. And then by the time you get beyond the child rearing years, we enter the calm, serene attachment phase, where you have enough hormonal stuff going on that you can tolerate your mate long enough to not only continue to participate in the raising of children who may now have reached the age of independence but are still around, but you can also tolerate your mate long enough to stay with him or her to the end, because in our society and in the human species, it's important to have the societal attachment in order to have people around you who can help to take care of you in your decrepitude, as the end of life so often is. So these systems are interconnected. One leads to the next. And Fisher says that if you don't want to fall in love with somebody, you probably ought not to copulate with them. She's not a moralist. She's not telling you don't have premarital sex. It's wrong. What she's saying is that because these different phases are interconnected, things occur when you secure a mate in the first phase that inevitably lead to the second phase, or have a tendency to accentuate the likelihood that the second phase and then the third phase will take place. So she's arguing that you're not necessarily in control, rather your hormones are. And that is because the system is set up to show this progression from one stage to the next. For instance, when you're in the testosterone or estrogen-driven lust phase, we know that the release of testosterone can also cause the release of the attraction hormones that will be important in the next phase, namely dopamine and norepinephrine. So if you're mentally stuck in the lust phase and you're just seeking, a bar hookup, your body might be doing other things that will help you progress to the next phase, or attraction, because your body, as a result of securing a hookup, may be going on to the next phase biochemically, which will lead you to that place behaviorally, and you start to fall in love. Or maybe you're just confusing sex with love, but sometimes they look quite a lot. And when you're talking about quite a lot the same, when you're using powerful hormones to regulate that behavior, you can definitely cross over a line that you don't intend to when you're thinking about it rationally. So repeated romantic encounters, that is to say sex, can cause a release of the other hormones, including ultimately the attachment hormone, oxytocin. And it turns out that semen from the male frequently contains hormones like dopamine. So if you're a lady and you are going to bars to find men and you're not really counting on falling in love, you're just out for a good time, but the semen provided by the male partner has dopamine in it, you might progress to that next phase all on your own. It's wicked, really. But it's the oxytocin that gets released that really seals your fate. Oxytocin levels will increase. This is usually late in the relationship, but oxytocin has other impacts, as we'll discuss in a moment. Oxytocin levels increase inevitably in a relationship that has been long-standing, and as a consequence, dopamine secretion decreases, so that reduces the romantic love with a feeling of calm, peace, and replacement of the passion that you once felt. Now, you might think this is a bad thing, and maybe you should get rid of that partner and seek a new one so you can go through the romantic attachment phase again, but in reality, it's probably good that you get to a place where you're not totally focused on one person all the time in sort of an obsessive compulsive sort of way, because if you were completely in that phase all the time, it would be difficult to have a job, it would be difficult to pay attention to your children because you're so focused on your loved one. So it really does make sense to tone it down on the romantic love side of things if you want to have an actual life and not just be crazy in love with one person who you think is the be-all and end-all. So that's why, biochemically speaking, you move on to another phase. And just to bring you up to speed on oxytocin, we've already said that it's the hallmark biochemical mediator in the third phase of love, but it also underscores many other adaptive behaviors. It is the ultimate social bonding hormone. It's a small little protein like the others we've discussed, consisting of 9 amino acids joined together. It's produced by a variety of organs, including the hypothalamus, the pituitary, and the ovaries, and most of the activity, although not all, but most of the activity is in women, and that's because it has a lot to do with successful reproduction. In fact, labor itself, if you're a pregnant lady, is started when oxytocin is released by the brain, and that initiates contractions in the uterus. In fact, if you don't go into labor on time, they will inject you with oxytocin to get the contractions started. At birth, oxytocin causes the contraction of the uterus to continue even after the baby and the placenta have been expelled to control bleeding. Birth is a massively bloodletting experience, and you need to control that. So we do it through oxytocin being released after birth. It also causes the letdown response in nursing mothers, without which it would be difficult to get the milk out of the human breast and into the infant's mouth. And so oxytocin facilitates that. But there's more. Oxytocin is absolutely critical for the bonding that takes place between the mother and the infant. And we know from experiments on rats and sheep that if oxytocin receptors are blocked in the female, she will abandon her babies. And also, if oxytocin is given to a virgin female who has not yet gotten pregnant, they will begin to exhibit spontaneous mothering behavior. So it appears as if oxytocin is intimately involved and very important for successful reproduction in mammals. Finally, and as we'll talk about in detail in the second section of the course, oxytocin induces social bonding, things that include monogamy and ultimately attachment. So, oxytocin, very important to human reproduction. All right, let's talk about some of the experimental evidence for the things I've just said about neurohormones, neurotransmitters in reproduction of mammals. There were 2 experiments done that really map this out very nicely and establish the absolute importance of oxytocin for bonding, both between babies and mothers, and also between the adults in the relationship. And this story in 2 parts, concerns 2 species of voles, little rodent-like animals. You see one little vole over here on the right. Very cute little thing. And so he will be one of the characters in our discussion here. Vole. And the first species of vole is known as the prairie vole. And to a certain extent, if you could compare the prairie vole to humans, you would conclude that the prairie vole is very much like the 1950s television program Leave It to Beaver, starring 2 iconic middle-class parents, namely Ward and June Cleaver. They were people who were just perfect models of what parents were supposed to be. Always there for the kids, the mother always had dinner on the table at 6 o'clock. The dad went to work, brought home the paycheck, mowed the lawn, all that stuff. So the prairie voles were similar in a vole-like way in the respect that the parents cohabited, they lived together, they jointly take care of the babies, and the male vole aggressively guards the female. So they were tightly pair-bound in a way that seemed very similar to the iconic 1950s human family. In contrast, however, we have the second species of vole called the montane vole, and they were precisely the opposite. There were no lasting pair bonds between the parents. They mated and then they parted ways. The males didn't stick around to protect the females, because he wasn't that interested, neither was she. And with respect to the female, care of the young was minimal. So a lot of the babies didn't make it, but who cares? Can always have another one. So we would describe the montane vole as promiscuous. They slept around a lot. They didn't care about having a single partner on whom they could account, and they were simply not loyal to any one particular partner. So we have a big contrast between the prairie vole and the montane vole. But here's what they did experimentally. First, the scientists took pair-bonded prairie voles and they gave them a drug that blocked their oxytocin receptors. Remember, we've been arguing that the pair-bonded animals did so because they were influenced by oxytocin. Well, when you block the oxytocin receptors, the mated pairs broke up. They couldn't get away from each other fast enough. We might go on to say that, they just sat on the front porch drinking cheap beer at that point, which obviously they did not. That's a metaphor. And here's some cheap beer, just in case you're wondering. Okay, so it looks like if you block the oxytocin receptors so that the oxytocin can't bind, then the behavior associated with oxytocin goes away. But then they also did the reciprocal experiment. They took the montane voles, the ones that were not pair bonded, and they injected them with oxytocin. And what do you know? Overnight, the montane voles became very models of propriety. And it didn't even matter if they were having sex or not. They didn't care. They just wanted to hang out with their mate and watch the sunset, maybe, sing romantic songs. Who knew? So the montane voles, who started out as sort of ne'er-do-wells, became monogamous pairs. And the males aggressively defended their female partner. So, tale of 2 voles, 1 hormone, 2 different results. And the complexity of this hormonally mediated behavior is evident in another study, this one conducted by Esch and Stefano. They too were interested in the neurobiology of love, and they looked at the underlying neurobiology of the different phases of love. Now, interestingly, they started with a definition of love, which Helen Fisher really didn't do. And they had two definitions. One was sort of a popular definition, which was that love is an attachment, intimacy, passion, grief upon separation, jealousy, in popular parlance. And then they also, because it's a scientific paper, they published a scientific definition of love, which was, love is a hypothetical and multidimensional construct with many interpretations and implications. And I think you will agree that that second definition really sucks. Well, maybe you won't agree, but to me, who'd want to be in love if that's what it meant? Anyway, the main point of the paper by Esch and Stefano is that love is a biochemical phenomenon and that the brain is the source of love. And they, like Fisher, argue that the brain is putting out neurotransmitters and neurohormones that influence the rest of the body. So love is simply the result of those biochemical interactions and the biochemical induced sense of yearning and subsequent satisfaction realized from being loved by the one that you particularly love. They also note, and this is really why I bring this paper up, that love is initially a stress-inducing phenomenon. It's really hard to put yourself out there, if only because most of us feel like we could be rejected, and that's very stress-inducing. So we need a powerful jolt of something, in this case, neurohormones, to get us out there or to get us to ask somebody out on a date. And the stress that I allude to shows up in those early encounters with the release of stress hormones called corticosteroids and in a positive feedback cycle. So once it gets started, once you start to release the corticosteroids, because it's a positive feedback cycle, more and more corticosteroids are released. And it takes a fairly significant countermeasure to overcome the stress-induced corticosteroids. But fortunately for us, our neurohormones can provide that needed trigger to get us to go out there to search for pleasure and the release from stress that we will enjoy once we find a partner and experience proximity and closeness. And indeed, if all goes well and the neurohormones do their thing, then once contact is made, the pleasure-inducing activities that cause the other hormones to be released will take over and create a sense of well-being. So this is their little diagram that kind of summarizes everything. Initially, we start out in a state of stress, both because we're afraid to go through the ordeal of possibly being rejected, and also this feeling of isolation because we're not with anybody, and that's, for most people, not the desired state. So eventually, we get to this point where we've got these corticosteroids going on, and in order to get us out there, we need at least some oxytocin, and if we can secure enough oxytocin to overcome the corticosteroids, according to these folks, Helen Fisher would probably disagree with this particular characterization, but that's why we have scientific papers, so we can argue over arcane facts. At any rate, because of the neurohormones, we go out, we find somebody, we begin the early stages of a relationship, and we may find this thing called love. However you want to define it. We're, for the most part, going to go with Helen Fisher's definition because she did a nice job of detailing the neurochemicals, and these guys did a less nice job of that, but they did do a good job of detailing the stress and the connection to corticosteroids. So that's the major thing I'm asking you to know from this graph, other than to also realize that there are multiple points at which you can have either a positive or a negative reaction, and these things operate in feedback cycles. right, and now we want to talk about the evolutionary significance of love and also relate that to hope, which is our ultimate goal in this section of the course. I've mentioned to you before that humans are born with excessively large brains. In fact, our heads are so big, our brains are so big, that we cannot be born with our head in their mature state. When you are born, the human head has to fit through a tunnel that is much, much smaller than the circumference of the head. And the only way to get the human head through the vagina is to make it malleable, to make it able to change shape. And that means not having the bones in the skull fused. If they were fused, they would not be able to change shape. And that would mean, most likely, that both the child and the mother would not survive the birth process. So rather than do that, what happens is that the human skull is not completely formed until you're about 18 years old. When you are born, it is highly malleable, and it can be made to change shape rather radically. In fact, if you've looked at a newborn baby, and I mean a neonate, within minutes of birth, they often look kind of like an alien because their heads are elongated and squished in ways that we wouldn't associate with a human head. And that's simply owing to the necessity of getting that baby and its very large head through a very small space and out into the real world. So this in turn means that because the kids are born with an incompletely fused skull, that they are born in an immature state, and their brains are not fully operational until a number of years later. This in turn means that children are born in a great state of dependency. And it requires a very high level of constant and committed parental investment to take care of that child and to make sure that it is not only cared for and protected, but shepherded through a series of behavioral suites and acquisitions so that the child is socially adept as well as able to feed and defend itself. So this training goes on for many, many years. This is one reason why we reproduce only one child at a time. It's such a huge investment of time and effort that it would be very difficult if we had a whole litter of kids like your typical dog or cat. Also, the reality is that we humans need to have at least 2 children per mated pair just to replace the parents. But if the population is to grow, we have to have more children than just those 2. And given the effort it is and the resource acquisition required to get children through to adulthood, humans typically don't have that many children. However, back in prehistory, it's highly likely that the human species itself teetered on the brink of extirpation on more than one occasion, because we are a slow-growing and slow-breeding species. We didn't have the capacity to grow the human population at a rapid rate for many, many millennia. Back in prehistory, as I said, we were probably at the point more than one time when a judiciously placed pandemic could have eliminated the entire human species from the planet. So, how do we keep people together long enough to go through this very lengthy, very trying period of raising children to independence? It takes a long period of time. Even if your options are limited, you still have to be compelled or enticed into staying with the same person for, let's say, 18 years, just for the sake of argument. So how do we do this? Well, it appears, if you believe Helen Fisher's arguments, that we do this by making the parents fall in love so that they stay together. And this in turn means that males will assume that protective role that helps keep the female protected from predators and other roving males. And it also benefits the male by assuring him that his genes will be passed on to his children and that they won't be somebody else's children. Similarly, there are attraction hormones in females that are stimulated by males who appear to be good providers. This means that the female can devote herself to the care and protection of her children, recognizing that because her male is a good provider, he will go out and do the hunting thing and perhaps some of the gathering, so that her children will likewise be protected and cared for. So hormones are terrifically important in mediating all of these responses. Another interesting note, and this is sort of by the by, is that in most animal species, once you get beyond your reproductive years, females are often just cast out and left to die. And the reason for that is, since they're no longer able to reproduce, they're basically drawing down resources without providing a compensatory benefit. And so in most animal species, those so-called non-reproductive or senescent females are almost always cast off. And without the protection of the group, then they'll be eaten by a predator or something like that. However, in humans, most human societies have been very good to senescent females. And this is probably because grandmothers play a pivotal role in teaching those really dependent offspring what they need to know about their culture in order to survive in an adult world. In addition, the grandmothers probably had some major role in the care of those young children, which would have released the reproducing females to do some gathering work, since they were, for the most part, hunter-gatherers at that time, and also to have more children to increase the population. Another interesting thought, and this is one that has been the subject of a book published by Catherine Coe called The Ancestress Hypothesis. Another suggestion is that grandmothers, or the ancestress in the title, were useful in that they begat art and invented culture. We know that humans are artists by nature. Every place we've seen humans on Earth, one of the things they've done is left some form of art, whether we're talking about cave art and drawing on cave walls, or pottery, or adorning weapons with patterns and things to make them look good. These are all evidence that humans have always been artists. Where did they get that propensity for art? Well, it may have been grandmothers who were overseeing all this. We know that one of the things that tribal humans needed was a way of identifying other members of their tribe. And they did that often by the use of body art. But how do they know where to find the materials to make the materials with which they would create the body art? Well, maybe that was an assignment given over to grandmothers who would go out and find the necessary materials, harvest the materials, amend it so that it could be useful, and then teach everybody the patterns and the icons that would uniquely identify a particular tribe. And these were not trivial undertakings. These were how people survived, how they knew stranger from friend, and so forth. And so if the grandmothers were doing this, even in their post-reproductive years, they were providing a vital function to human society. More recently, we've come up with some evidence that, at least among some primates, where we'd expect at least some nascent human behaviors to be visible, that perhaps the Ancestress Hypothesis has a broader reach than just Homo sapiens. This is a story about two grandmother monkeys who were seen intervening to raise their own grandchildren after something happened to the mothers. It's not clear whether the mothers simply took off because they were no longer interested or whether they were killed in some way. But in any event, the grandmothers took over and apparently were able to re-achieve the ability to produce milk for the young growing children. In another study, this one on macaque grandmothers, they found them nurturing granddaughters when mothers took off. We have no other records, extant records, of senescent females doing this outside of human beings. But it looks as though in our species and others, sometimes grandmothers did intervene if they had dependent offspring with no mother to take care of them. All right, I promised to discuss the relationship between love and hope. What might that be? Well, I would argue that hope underpins the whole concept of love, both in the seeking of it and the sustaining of love. Love is, if we go with Fisher, a motivational system, and hope is a key motivator. As it turns out, hope operates on the same neurochemicals as does love, coming from many of the same organs. And it's a bit simpler to understand. So, we'll go with that. Now, do we know anything about the neurobiology of hope? And the answer is, yes, we do. Here's a very short article called The Neurological Basis of Optimism that was published in the Curious Neuron back in 2012. And it's a study that looks at people who are self-professed optimists versus pessimists. And the key distinction between these two groups is that they draw very different conclusions and take different actions and also suffer different vulnerability to stress based on the same information. So, being able to make these distinctions about whether you're an optimist or a pessimist would seem to have some utility in determining what kind of a mate somebody would be, what kind of an employee somebody would be, what kind of work colleague somebody would be. And we would also like to know if there's a key difference in the brain of those people who are testing as optimists versus those who are pessimists. Can we do any of that? And the answer is yes. Here's an example of a scientific study coming from studies using fMRI, functional magnetic resonance imaging, on people doing some behavioral tasks. And they were specifically investigating this concept of optimism bias, which is a brain state in which the individual who has optimism bias overestimates the chances of a positive, beneficial outcome, even though there is factual evidence to the contrary. So even though you're looking at evidence that suggests you're not going to do very well, nonetheless, internally, your brain is busy overestimating your chances of success and feeding you information that you're going to be really successful, even though the data says, no, this isn't going to work. And we know biochemically that we do this, and we're not the only species that does this. But we do this because we are spurred by the expectation of a reward. And you remember that the amygdala is a place where rewards are processed, and it appears that the amygdala is operating in optimism bias, and that it sends the brain messages that result in dopamine secretion. You will come to know dopamine as the I feel good hormone. So here's an experiment that they conducted to see if they could uncover more about the biochemical nature of hope, expectation, and optimism. They asked subjects to choose from a list of vacation destinations. So they had a list of places you could go on vacation, like Florida or Alaska or Colorado or wherever. And the participants were asked where they would be happier on their next vacation. After that, they administered L-DOPA to the participants. L-DOPA gets converted into dopamine inside the human body. And what they found was that after they got the L-DOPA, there was an enhancement of the preference of their choice. So if they'd chosen, I want to go to Denver, Colorado as my choice, after receiving the injection of L-DOPA, they were really, really focused and really, really excited about going to Denver, Colorado as a result of the L-DOPA injection. They also noted that there was differential processing of positive versus negative information. So once they chose Denver, Colorado, they would feed them either positive or negative information about Denver, Colorado. And what they found was that positive information was mediated by the amygdala, which they determined from the fMRIs, and negative information was mediated by the cingulate cortex, where errors are processed. So they suggested that one thing that came out of the study was that perhaps depression could be due to the fact that the cortex overrides the amygdala and suppresses the positive information. But they concluded that the natural state for a normal human is optimism, and that optimism itself is an adaptive trait. And that's probably true. When people say you can't live without hope, they're really talking about optimism. And to the extent that optimism is a biochemical state, it is thus natural. As an aside, they also found evidence for optimism bias in other species, such as rats, dogs, rhesus monkeys, and birds, starlings. But the birds were more likely to interpret ambiguous results as optimism. So maybe birds are super optimistic. You know, when it's iffy, they go with the optimism. All right, and here's your study guide and what you need to know for module on the biochemistry of love.